AppSec's online support program, Class 9, Week 9, Lesson 4, Subject Biology. Textbook Biology, page number 89 to 91, Unit 4, Cell Cycle, topic Mitosis. Objectives. By the end of the lesson, students will be able to revise mitosis, phases of mitosis that are karyokinesis and cytokinesis, then phases of karyokinesis that are prophase, metaphase, anaphase and telophase. Cytokinesis, then how cytokinesis take place in animal cells and then in plant cells. Mitosis. First we will be discussing the discovery of the mitosis. In 1880s, a German biologist, Walter Fleming, observed that in a dividing cell, nucleus passes through a series of changes which he called mitosis. Now, the definition of the mitosis. Mitosis is a type of cell division in which a cell divides into two daughter cells, each with the same number of chromosomes as were present in the parent cell. Mitosis only take place in eukaryotic cells. Now, a question arises, why mitosis take place only in the eukaryotic cell? Because eukaryotic cells are provided with a proper nucleus. In multicellular organisms, the somatic cells undergo mitosis. Now, in which cells mitosis take place? Mitosis take place in the somatic cells. What are the somatic cells? Somatic cells are the one which make the body. Phases of mitosis. There are two major phases of mitosis, that are karyokinesis and cytokinesis. What is meant by karyokinesis? Karyo means nucleus, kinesis means division. So, karyokinesis means the division of the nucleus. What does the term cytokinesis mean? Cyto means cytoplasm, kinesis means division. So, it is the division of the cytoplasm. In mitosis, remember that first the division of the nucleus will take place and then the division of the cytoplasm will take place. Now we are having some phases of the karyokinesis too, that are prophase, metaphase, anaphase and telophase. Now we will be discussing these phases in detail one by one. Prophase. Prophase is the first phase of mitosis and it is the longest phase of mitosis. Here, genetic material is in the nucleus is in a loose thread-like form called chromatin. So, the genetic material will be present in a Maggi noodle-like form, and that Maggi noodle-like form of the genetic material actually called chromatin. Then, the chromatin will arrange itself in a highly ordered structure which will be called as chromosomes. Chromosomes are actually having the two sister chromatids and they are attached at a point of centromere. Around the centromere, kinetochore will be present. What are kinetochore? Kinetochore are actually the proteins and which help in the attachment of the spindle fibers. Now, there are two centrioles that are close to the nucleus and the two centrioles are collectively called the centrosomes. Now these centrosomes will duplicate itself and they will move to the opposite poles. So both the centrosomes migrate to the opposite poles of the cells. Now from the centrosomes there arises microtubules. These microtubules will join to the tubulin proteins. As you all know that tubulin proteins help in the formation of the spindle fibers. Now the spindle fibers are being formed. When the spindle fibers are formed, so the complete set of the spindle fiber will be known as mitotic spindles. At the end, nucleolus and nuclear envelope will also disappear. Now we will be discussing the prophase in plant cell. Like you all know that the plant cells are also having the vacuole and they are having the nucleus at the site. So in highly vacuolated plant cells, nucleus has to migrate to the center of the cell before prophase. Before starting of the prophase, when the cell decides that it should be divided, so the nucleus will come to the center. The cells of the plants lack centriole. Now, if the plant cell is lacking centriole, so here a question arises that how spindle fibers will be formed. So, the spindle fibers are formed by the aggregation of the tubulin proteins on the surface of the nuclear envelope during prophase. They are not having centrioles, but they are having the tubulin proteins, and tubulin proteins actually help in the formation of the spindle fibers. So, they will join together on the surface of the nuclear envelope and thus spindle fibers will be formed. You can here see the structure of the prophase, right? We are having the chromosomes, right, that are having the two sister chromatids and they are attached at the point of centromere, and around the centromere kinetochore proteins are also present. You can see the nuclear envelope too, right, and the two centrosomes are also present which are close to the nuclear envelope, and from the nucleus you can see that the mitotic spindles are arised. Second phase of mitosis is metaphase. Simply, what does metaphase mean? Metaphase means that the chromosomes will come to the center, they will align themselves in a line. When spindle fibers have grown to the sufficient length, some spindle fibers known as kinetochore fibers attaches with the kinetochores of the chromosomes. As I have already told you that what is the function of the spindle fiber? The function of the spindle fiber is that they attach to the chromosomes at the point of the kinetochore where the kinetochore proteins are present. They help in the attachment of the chromosome, right? Two kinetochore fibers from opposite poles attached with each chromosomes. I have already told you that the centrosomes will move to the opposite poles, and from the centrosomes spindle fibers are arised, and those spindle fibers are attached at the point of the centromere of the chromosomes. Now the chromosomes arrange themselves along the equator of the cells, forming metaphase plate. You can see in the diagram that the chromosomes have arranged themselves in a line, and the spindle fibers have been arised from the centrosome and they attaches itself at the point of centromere in each of the chromosomes. Third phase, that is the anaphase. Actually, what happened in the anaphase? Here, the pulling of the chromosomes take place. You can see when a kinetochore spindle fibers connects with the kinetochore of the chromosomes, it starts pulling. As in the metaphase, you can see that the spindle fibers has attaches itself to the chromosomes at the point of centromere. Now, when they attaches itself, so what will happen? They will start pulling in the opposite direction. The pulling force divides the chromosomes, sister chromatids, and they are pulled towards the respective centrosomes. Okay, so when the pulling is started, so the chromosome will be divided into the sister chromatids and they will be pulled towards the respective centrosomes, as the centrosomes are present on the opposite poles. The other spindle fibers, the non-kinetochore, also elongate. Now, what are the non-kinetochore? Non-kinetochore spindle fibers are those which do not take part in the attachment of the chromosomes, or the spindle fibers which are not attached to the chromosomes, they are actually called as non-kinetochore. And in anaphase, the non-kinetochore spindle fibers will be elongated. At the end, cells has succeeded in separating identical copies of chromosomes into two groups at the opposite pole. Now, as I have told you that the chromosome consists of two sister chromatids. When the pulling is started, so the chromatids will be detached. Half will move towards the opposite pole and half will move to the another opposite pole, and thus two identical copies have been produced. You can see the diagram of the anaphase. You can see that with the help of the spindle fibers, the chromosomes have been moved to the opposite pole. They are now detached, right? They are now detached. They are called the sister chromatids now. Another phase of mitosis is telophase. Remember students that telophase is the reversal of the prophase. As we have studied in prophase that the nucleolus disappear, the nuclear envelope disappear. The genetic material is present in a loose thread-like structure called chromatin. The centrioles duplicate itself into two centrosomes. They move to the opposite pole. So, telophase is actually the reversal of the prophase. What will happen in the telophase? A new clear envelope formed around each set of separated chromosomes. As we have studied in the prophase that the nuclear envelope disappear, but in the telophase the nuclear envelope will be again formed around each set of separated chromosomes. As in the anaphase, I have told you that the chromosomes are moved apart to the opposite poles in the form of the sister chromatids. So, from that sister chromatids actually a nuclear envelope will be formed. Both sets of chromosomes now surrounded by new nuclear envelopes unfold back into chromatin. Now, when the nuclear envelope is being surrounded by each set of separated chromosome, so they will be again converted into the chromatin. Then the nuclear division is completed at this stage. At telophase stage, the nuclear division is completed. Why the nuclear division is completed? Nuclear division is completed because the nuclear envelope has been formed from each set of the chromosomes, and the chromosomes will again converted into the chromatin. You can see the diagram of the telophase, right? From each set of the chromosomes, a nuclear envelope has been formed, and each cell is also provided with one centrosome also. Now, cytokinesis. It is the division of the cytoplasm. As I have told you earlier that cyto means cytoplasm, kinesis means the division of the cytoplasm. Now, how cytokinesis takes place in animal cells? Cytokinesis occurs by a process known as cleavage. A cleavage furrow develops where the metaphase plate used to be. The furrow deepens and eventually pinches the parent cell into two daughter cells. So, in cytokinesis, simply the cytoplasm will be divided and the two daughter cells will be formed. You can see the diagram. As in the diagram it is shown that the cytoplasm is getting divided. So, when the cytoplasm is divided, so the two daughter cells will be formed, and each having the same number of the chromosomes as we're having in the parent cell. Now, the cytokinesis in plant cell is somewhat different. It is also the division of the cytoplasm, but in plant cells, vesicles derived from the Golgi apparatus, as you all know that vesicles are formed from the Golgi apparatus. So, that Golgi apparatus will move to the middle of the cells and fuse to form a membrane bounded disc called cell plate or phragmoblast. So, the Golgi apparatus actually move to the middle and they will be fusing with the membrane bounded disc. When they fuse, they will be forming a plate that is called the phragmoblast. The plate grows outward and move vesicles fuse with it. Finally, membrane of cell plate fuse with the plasma membrane and it contents joined the parental cell wall, and it helps in the formation of the two daughter cells. Here you can see the vesicles these vesicles are fusing and they are forming a membrane bounded disc that is called a cell plate, or it is called a phragmoblast. You can see over here in the next diagram that the vesicles are fused and they form a plate like structure, and when they form a plate like structure, so with the help of the phragmoblast, the two cells will be separated from one another and two daughter cells will be formed. So students, this was your today's lecture. Hope you find it easy and interesting. Thank you.